Hello, dear student. Myself, Dr. Lakshman Rathod. I am working as assistant professor in Department of Botany, Mahatma Phule Art, Science and Commerce College, Panvel. Okay. Our today's topic is Tikka disease of groundnuts. Okay. First, groundnut plant, botanical name, Arachis hypoxia, pathogen, Cercospora personata, and Cercospora arachidicola. It is a major disease of groundnut in India. Tikka is one of the most destructive disease of groundnut. The disease is commonly in USA, that is United States America, Philippines, Africa, Indonesia, Australia, and India also there. Okay. So when the disease takes place in the field of groundnut here, the 15 to 50% have been reported due to the disease incidence. Means what? 15 to 50% losses in the agricultural fields also. Okay, you can see the symptoms here, dear students. The you can see different type pictures of the infected part of the groundnut. Okay, so first is a leaf spot here, Cercospora personata. Second figure shows the Cercospora arachidicola, and third part shows the infected groundnut pod with disease. Okay, and uh, fourth figure shows the infected groundnut stem with disease. Okay, you can see the first figure, the student here. Firstly, the symptoms which occur on the surface of leaf. Is it clear? Then, and the first spot shows means first figure shows the Cercospora personata species, which is smaller in size, surrounded by yellow color, pale yellow color here. Then, uh, while in a second figure, the disease takes place on the surface of leaf. The spots are irregular in size. It is a large in size. Now, finally, the disease are slowly spread to the stem, and then finally seed also there, or pod also. Okay, so that is a simple way of the symptoms. When the disease are takes place on the both surface, actually the symptoms seen in the month, up, after 2 months. The symptoms are seen in after 2 months or 2 month old of the plants. Then the actually disease are takes place. That is about symptoms. Now, dear students, about causal organism, the disease is caused by fungi, Cercospora personata and Cercospora arachidicola. Okay, that is a two types of spots which called the Tikka disease of groundnut. Tikka disease is also known as a leaf spot disease. Okay, then about mycelium. Mycelium is a well developed branch, septate, cylinder, intercellular, and brown in color. Means what here? Mycelium means what? A bunch of group of hyphae is known as a mycelium. Before the formation of spot, the myceliums are present on the surface of the. Then the slowly mycelium spread, their formation, reproduction there. After the reproduction, the conidia are formed. Then the conidia germinate into the new thallus. That is a simple regarding the uh, mycelium. Okay, then branched hostorium are present or absent in absent in Cercospora personata. Hostorium means what? Hostorium is one type of food sucking organ. It is a knot like structure. Okay, the hostorium present on the surface of leaf. From with the help of hostorium, the hostoria sucking food organ and provide to the entire thallus of the plants. Okay, that is a simple way regarding mycelium. Now, dear students, you can see the asexual reproductions. So actually, in general, in fungi, three type of reproduction or two type of reproduction is there: sexual and asexual reproduction. Is it clear? But in this group, only asexual reproduction is there. Why is asexual reproduction? Because the fungi is imperfectly here on the lack of sexual reproductions. Is it clear? That is a only simple way asexual reproduction. The asexual reproduction takes place by asexual reproduction takes place by formation of conidia. You can see the figure of conidia. Okay, here mycelium, the mycelium which are present on the epidermal cell of leaf. Here you can see the infected tissue, that is a infected host tissue. Then at the base of the mycelium, here small, you can see the um, small knot like structure. Okay, that is a hostorium or mycelium. Okay, the mycelium enter inside the leaf tissue, and from there the mycelium sucking our hostorium sucking food material and provide to the entire thallus structure. Okay, then the asexual reproduction takes place by formation of conidia. Where is conidia presents? The conidia are present on the conidiophore. And where is present the conidiophore? The conidiophore are present on the surface of the or on the mycelium. The mycelium are present on the surface of leaf. The conidia are septate here. It is elongated. Okay. Then at the tip of conidiophore, the small chain-like arrangement, that is conidia. Okay. At that at the maturity stage, the conidia are separated from the conidiophore and they can carried by wind in the favorable condition. While in the favorable means what? When the conidia are sufficient, provide food material, sufficient available climate is there. Okay, then the conidia are germinate into the new thallus, or they can cause the diseases. That is a simple way of a sexual reproduction. Is it clear? Then about disease cycle. Ata how is disease spread? Here, first is a penetration, second primary infection. Okay, the disease perpetuates through conidia laying in the soil on diseased plant debris and through the conidia being carried in the cell of groundnuts. Okay, Perenation is one type of penetration part. How the disease are actually takes place? Actually, the disease is soil-borne pathogens. Okay, what is soil-borne pathogen? The pathogen which are present inside the soil. Okay, then uh, in the soil, the pathogen is takes place when the groundnut are so in the soil. Okay, then after the 2 months, after 2 months, the pathogens are slowly active and they cause they enter on the they are present on the surface of leaf and they are slowly spread to the diseases. Okay, then another primary infection. When the new crop of groundnut start growing, the viable conidia are brought to the host of surface by various agencies, germination in favorable condition and cause primary infection. Then after the fully growth of the plants 2 to 3 months, then the pathogen are active and slowly on the present on the leaf. Then the from the leaf the pathogens are entry into the stem part. Then after the stem part the pathogens are spread to the cell or even the pods. Okay, then finally disease takes place. That is the disease cycle. How is spread the disease? First of all, the pathogen is present inside the soil. Then the pathogen has penetrate into the plants, or they are present on the surface of leaf. Then after that, after then, the pathogens are spread on the stem surface. Finally, they reach to the cell. Even though they are cause the root also. Okay, then finally they cause the pods. Okay, pods is actually present inside the soil. Is it clear? So that is about disease cycle. You can see the one type of cycle, active phase, two type of phase here, active phase and dormant phase. In active phase, myceliums are active, they cause the host infection. Finally, there's further spreading of the infected by conidia. The conidia germinate, then germinate. In active phase, the conidia only germinate. While in dormant phase, the penetration as conidia on the plant debris, on the cell, on the seed. In dormant phase, the conidia will not germinate. While in the favorable condition, means what? In active phase, the conidia germinate to cause the diseases. Okay. And last is the important part, control measures. Okay. Control measure is a very important part in the diseases of in the disease. Here in this control, as you seen in a chemical control, biological control, and physical controls. Okay, from which here we have to see some points are highlighted here. Then first of all, there are three types of control measure. Actually, there are generally three types of control measure. As you seen before previous lectures in physical control, biological control, and chemical control. Okay, cultural practice is there. Cultural practice is nothing but one type of physical methods. Okay, then the plant debris from the previous crop should be burned to avoid the soil-borne primary infection. Means what? When uh, after the harvesting stage or during the harvesting stage, the highly severe infected plant is there. Okay, such type of infected plants are uh, uh, collected and uh, uprooted from the soil, and the plants are burned through the field or uh, through the fields. Okay, so then second, rotation and deep burying of debris may be also help to destroy the soil borne pathogens. Is it clear? Rotation is important parts. Okay, crop rotation is there. Crop rotation is very important aspect regarding these issues. Here for example, uh, in one crop rotation, actually 3 to 4 year the crop rotation is very necessary. Okay, for example, in 1 year we can grow the crop, soybean crop is also there in field. Is it clear? Then in next year we cannot grow the same type of crop. We can grow the alternate crop, that is jawar is there, alternate crop, ground another uh, moog is there, that is facilus, and udid is there. Okay, so that is a regarding crop rotation. So then another part is a alternation in the date of sowing can be help to avoiding the damage of disease. Alternate sowing is very important. Okay, then biological control, biological methods. Here in these methods there are different microorganisms, non-pathogen organisms. So the biopest is there, which is prepared by microorganism. The microorganisms are applied to the seed or the plants. Here the Cercospora and Personata and Arachidocoda are controlled by using these methods. Now another biological control is there, foliar application of aqueous neem extract, neem seed kernel extract at 2 weeks interval 3 times starting from 4 weeks. Okay, for after planting is good. So here neem cake is one type of prepared by neem Azadirachta indica plants. Okay, the paste are useful to control the leaf spot of our ticka disease of groundnuts. Now last is their chemical controls. So the seed treatment is essential to eliminate seed-borne inoculum. Before the sowing period, we have to apply some fungicides or chemicals to the seed. Then the seeds are kept in the light uh, 15 to 20 minutes or half an hour. Then the seeds the seeds can be grown. Then that is a to avoid the chances of infection. Next is there, infection of seed with cell can be done by using sulfuric acid, another copper sulfate solution, ergosan, Bordeaux mixture, diathenium 45 and carbon diazine. These all are the some fungicides are powder form. Or then these all this fungicides are chemicals which use to control the plant to the ticka disease of groundnuts. Okay, that is simple way of control measure. And lastly, thank you.